Now, what I'm going to present is an investigation study of a method that has been first introduced by Grossman, as we will see in a minute. How the presentation is going to be structured: we will have an introduction on what are the electric stimulation methods and what this new method is about. Then we will see what this work is about and what are the possible future steps. And also, there's an invitation for poster number 36 later today in the poster session, and then you can see the references always in the presentation. Now, regarding the electric stimulation methods, these are widely used and they date back to the 20th century for transcranial magnetic stimulation and transcranial electric stimulation, and they are used for a purpose to actually try and stimulate the brain without opening it. Now, TDCS, which is transcranial direct current stimulation, is the most well-known method and has been used for the treatment of depression, and is also FDA approved, and has also been used you can find in literature for other conditions. Although there are more studies need to be made there to draw conclusions on the effectiveness of this method, that you can see those references in this presentation later. Another method is the TACS, which utilizes the same mechanisms, but now we're talking about alternating current, and the frequency factor plays a role now. So if the frequency is low enough, we can have neuromodulation. So in TACS, we are opting for like 10 or 40 40 hertz, depending on what circuit we're looking for. Most of those methods lack focality, and you can see in this picture that only 2 electrodes were active, and you can see how sparse is the stimulation. And thinking about deep brain stimulation, where we want targeted pointed stimulation inside the brain, that's a no-no for those methods, TACS and TDCS, although they can be effective for larger networks. The new method is the transcranial interference stimulation, or something like that, which is introduced by Grossman at 2017. This is a promising method and has been tested on murine subjects and has shown that it works. And as always, as we said before, everything works in murine models, but when it comes to humans, it's a different story. And it achieves a great focality and penetration depth. This is a simulation. This is an actual simulation of the human brain model from the PHM repository of the ITIS Foundation, and this is actually one figure from this work. And after the study, after the first introduction in 2017 from Grossman, there were some other studies from Rampersad, for example, in 2019, showing this method on human models. Why do we do TTIS now? TTIS is used to is a promising technique, as I said, because it can go deep and it can be pointy. We can use it in non-invasive deep brain simulation and achieve the pattern steerability and focality we like. As we will say later, and you can also ask more in the poster session and have an interaction there. It depends on very much on where the electrodes are placed and how actually you're steering the beam in order to achieve the focality. One great thing that this method provides is that if someone sees it from the TACS perspective, we can see that this method, it's not focal at all, but when combined, actually what we combine, we'll see later, it becomes focal. How this method works, actually two electrode pairs are used on the contralateral sides. Of course, the position matters, and the ejected frequency of each electrode pair differs slightly, just to create enough envelope in order to cause the modulation we want. Interference is used, and as you all know from high school, probably you have all seen this graph. when two sine waves are superimposed, you can create this interference pattern, and that is what this method is utilizing. If we see each sine wave and the pattern that causes on the brain, then we see an image same as this one. But when we combine those, the image becomes like this. We don't care about the image that is like that, because the each of sine waves are in the kilohertz range of frequencies, much higher sometimes than the cutoff limit of the neurons, so they don't modulate or they don't cause any problems, at least what it shows to be in the surroundings. What has been shown for the human models, as you can see this picture, is that for the same placement, we use here the 1020 electrode system, that the same that is used for the electroencephalography studies. For the same electrodes, we can see varying patterns. That is a bad thing. So that means we need a personalized treatment, at least if we are going to use the method. Also, you can see here, the same is actually, I will show the axis later, is across one line, the variation of the electric field along with the standard deviation of each one. And this is across the center of bounds of the human brain. We will see, like the first diagram was on this axis on the left, and the other was on the right. Now, what are the goals of this work and the future work? Actually, the optimization is required, as you can see, as you saw in the pictures there. We need to optimize and try to find the best electrode combination, and also the best potential combination for the electrodes, in order to be able to achieve the focality and the pattern we want. The problem is that the problem is nonlinear and non-convex. That means, well, Houston, we have a problem here with the optimization. And as far as currently now, the work, this is an ongoing work, and these results have shown to be a bit difficult to optimize for specific targets, and they were only tested on 1020 electrode system. If a greater like more electrodes are used, maybe the we will have a convergence, hopefully. Optimization has already been tried by Rampersad, but this was a kind of brute force way to approach the problem. Here in this work, we're trying with genetic algorithms, or we're at least trying to find which algorithms can achieve this optimization. Finally, to try and reach a personalized optimization, hopefully getting the MRI data, then segmenting it and trying to find the desired pattern based on which area we want to modulate. You're all invited in this poster session. The poster looks like this. It's number 36. And yeah, the usual slides. This presentation is open, and you can all see it on my GitLab repo there. Also, the poster is there, and that was it. Thank you for your attention. And if you want to check the references, you can always go to the presentation and see the references here.